Cote here. Welcome to the second video on the immune system. In the last video, we looked at innate immunity, the nonspecific immunity that we have from birth. Now let's look at adaptive immunity, focusing on B cells and T cells and how they develop. So let's get started. Adaptive immunity is something that's unique to vertebrates. It can also be referred to as acquired immunity because this is the type of immunity that you acquire or get over your lifetime. The two main types of white blood cells that are involved are called lymphocytes. These are the B cells and T cells. All blood cells start off in the bone marrow. The ones that stay and mature there are called the B cells. That's why they're called B cells. Others travel and mature in the thymus, and these are appropriately named the T cells. So that's what the B and the T stand for. Pretty simple naming, right? One very important concept in adaptive immunity is that the B cells and T cells only respond to pieces of pathogens called antigens. When an immune cell sees something hanging out in your body that shouldn't be there, it engulfs it, rips it to pieces, and usually displays a piece, what we refer to as the antigen, for the rest of the immune system to see. These antigens will bind to specific receptors on the responding immune cells and activate a response. To be more accurate, let's look at this diagram, and this is not in your book, by the way. It's something I found that gets the point across. This whole piece would be considered an antigen, but notice how this antigen has different shapes coming off of it. These specific shapes are called epitopes, and these are what will actually bind to the receptor. So now that we know what the bad guy looks like, let's focus on the good guys, the lymphocytes. Here's a picture of a mature B cell and a mature T cell. The purple pieces coming off of them are their receptors. Notice that B cell receptors have a Y shape to them, while the T cell receptors are straight. The tips of the receptors are what would have the specificity to recognize the different antigens. So let's take a closer look at these receptors. Each B cell receptor consists of 4 polypeptide chains, 2 identical heavy chains, the longer ones on the inside, and 2 identical light chains, the shorter ones on the outside. These chains are held together by disulfide bridges, shown in yellow. Remember, these are when 2 sulfurs form a bond with each other, most likely from 2 amino acids with sulfur in their R groups being near each other. Notice that on each chain there is a C region and a V region. The C stands for constant, which means that this part will be the same between the 2 chains. The V stands for variable, and this is the part that's different between the chains. This will ultimately give the tip of the receptor its variation, making it specific for an antigen. You have around 1 million different B cells in your body, each specific for a different antigen. Looking at a T cell receptor in more detail, we can see that there are only 2 polypeptide chains involved, an alpha and a beta chain that are linked by a disulfide bridge. These chains also have a constant and variable region to produce variation, and you have around 10 million different T cells in your body. So while B cells and T cells can bind to antigens, they work in different ways. We're going to look in more detail at what happens when these cells are activated in the next video. But right now, it's very important that we take a look at how these cells are developed in the first place. There are 4 main characteristics in building an adaptive immune system. First, you need to create receptor diversity in your B cells and T cells, so they'll be able to recognize the wide variety of pathogens that are trying to invade you and get you sick. You also need to make sure that these cells that you're going to let loose in your body have the ability to show self-tolerance, to be able to distinguish between self and non-self. If your immune cells react to and attack self, this will lead to autoimmune disorders, where your lymphocytes are attacking your own cells. And this is not good. When activated by an antigen, the cells need to proliferate or make a lot of copies of itself, since this is the cell that recognizes the pathogen. And last, you need to reserve some of these activated cells, called memory cells, so that if the same pathogen ever tries to attack again, you already know how to handle it. So let's go through each of these steps using B cells as an example. Remember that receptors are made from polypeptide chains, and polypeptides are made from the transcription and translation of DNA. So how do we get 1 million different B cells and 10 million different T cells? The answer is that we can produce different combinations of a gene from recombining the original DNA strand. For simplicity, this figure shows how one light chain is made for a B cell receptor. The light chain is made by putting together 3 gene segments: a V segment, a C segment, and a J segment. J stands for joining, by the way. Looking at the top strand of DNA, all the way to the right is the C segment. Remember, this is constant, so there's only one possible gene segment for this. All the way to the left, there are the V segments. Remember, this is for the variable region, and while they don't show all of them, there are 40 V segments in the light chain DNA. There are also 5 J segments in between the V's and the C. So what happens is that an enzyme complex called recombinase will randomly link one of the 40 V segments to one of the 5 J segments. This can give you 200 possible combinations. 40 times 5 is 200, get it? In this example, it combines V39 and J5. This gene will then be transcribed, processed, and translated into what will become the light chain. Understand that for the heavy chain, in addition to the V, J, and C segments, there's also a D segment, making way more possibilities for diversity. And this is how we get millions of cells, each having a unique receptor shape. Because the receptors are made through a random process, sometimes they'll be able to bind to proteins on our own cells, and we don't want that to happen. So before they fully mature and be released into the body, they need to undergo a final exam called self-tolerance. Basically, they are shown an epitope from our own cell, and if they bind and react, then they'll need to be destroyed by apoptosis. The ones that pass the test will be allowed to go out and find foreign molecules. So after the B cells graduate from self-tolerance, they go out and they just wait until they match up with an antigen. Here we have 3 different B cells, each with a different receptor. The antigen at the moment are the orange pieces, and you can see that only one B cell, the middle one, has the right receptor that matches an epitope on this antigen. For that particular antigen, the B cell with the purple receptors is the one that will be activated. The other 2 are not destroyed, and they'll just wait around. Who knows? Maybe one of them will match with the next pathogen that comes along. After activation, the chosen B cell needs to divide to make many copies of itself, and this is otherwise known as proliferation. Since the copies are identical, this process can also be called clonal selection. The clones are then divided into 2 groups. Some cells will become plasma cells. They will go out and fight the good fight by secreting antibodies specific to the antigen to neutralize them. The other cells become memory cells. These cells are not actively fighting. They will live in the body for a long time, and if the same pathogen tries to invade the cell again, they will remember how to fight it off. The pathogen will be taken care of before it has time to do anything. Which brings us to the last of the characteristics, memory. This is how you acquire your immune system over your lifetime. Whenever you encounter a pathogen and develop a response to it, you're also creating memory for that specific pathogen. So over time, you can build quite a library of memory. The first time your body comes across a pathogen, this is termed the primary immune response. This is when a specific B cell or T cell is activated and undergoes proliferation to make the 2 types of clones. This is a relatively slow process and takes some time. However, because of memory cells, if your body sees the same pathogen in the future, the memory cells launch into a secondary immune response, which is much quicker because it already knows what to do. Here is a visual representation of what the primary and secondary responses look like. The blue line represents the primary immune response to pathogen A. As you can see, the first time around, it takes a while, a few weeks, to figure out how to eventually destroy A. But look at what happens on day 28. When you are exposed to both pathogen A and pathogen B, the response to A is very quick because you've already seen A before and you have memory cells and you know how to get rid of it. And since you have never seen pathogen B and don't have memory to B, you need to start from scratch and create a primary response to B, which is represented by the red line. So every time you get sick or get a vaccine, this is what's happening in your adaptive immune system. You're creating memory. So this is where I'm going to stop for this video. Hopefully you've learned a little bit more about B and T cells and how they develop. In the next video, now that we know the basics, we'll explore the specific responses of B cells and T cells because they do act differently in the body. So until next time, be smart and be safe.